Hello, and thank you for joining us. My name's Rachel Guberman-Hill. I'm Director of the Elizabeth Blackwell Institute for Health Research at the University of Bristol in the UK. I'm here to welcome you to the 2021 Elizabeth Blackwell Institute lecture. Thanks for joining us from wherever you are around the world. This lecture will be given by Professor Dame Sarah Gilbert. The Elizabeth Blackwell Institute takes place every year to honour the spirit of Dr. Elizabeth Blackwell, who was born 200 years ago. And so, as a very short introduction, I'd just like to say a few words about Elizabeth Blackwell herself. Dr. Elizabeth Blackwell was born in Bristol, where I'm speaking to you from now. She became the first woman to be awarded a medical degree in the United States of America. After being qualified, she returned to the United Kingdom, and in 1859 she received her place on the British Medical Register. Her place on the medical register meant that she was able to practice as a doctor in the UK, and this means that many people describe her as the very first woman doctor. Dr. Blackwell worked really hard to prevent illness, to improve health, and to provide medical care. She also ran public lectures to inform people about prevention of illness and to stimulate discussion about health. Her work inspired others to follow in her footsteps and to become doctors and scientists. Today's lecture echoes that pioneering spirit and innovation that Dr. Blackwell embodied herself. So I'm really honoured that the 2021 Elizabeth Blackwell Lecture will be given by Professor Dame Sarah Gilbert. Sarah holds the Said Professorship of Vaccinology at the Jenner Institute at the Nuffield Department of Medicine in the University of Oxford. She needs very little introduction for her work to develop the Oxford AstraZeneca COVID-19 vaccine. Today, Sarah is going to take us on a fascinating journey through how the vaccine was developed. The lecture itself will be about half an hour in length, and she'll then answer some questions from you, the audience, that you've very kindly sent to us in advance of today. Sarah, thank you for joining us for the Elizabeth Blackwell Institute lecture in the year that marks 200 years since Dr. Blackwell was born. I'll pass the microphone over to you now.
Thank you very much. I'm very pleased to be here with you today to give this lecture. So I'm going to take you through the story of what we did starting at the beginning of 2020 and finishing up with where we are more recently in the development and deployment of the vaccine that we developed in partnership with AstraZeneca. So first I want to spend a few minutes just describing the technology that we use. And this is really important because the reason we were able to go so quickly, or one of the main reasons we could go so quickly in 2020, was that we use what's called platform technology to develop a new vaccine. And that is important because it means we do a lot of work on the vaccine platform itself before we actually start to use it to develop a vaccine against a specific disease. And that means that because so much of the work has already been done, when we decide which disease we want to use it to make a vaccine against, we can proceed very quickly because we've already answered so many of the questions that need to be answered before we can start to use the vaccine. We've already worked out how to produce the vaccine, how to test it prior to going into clinical trials, how to manufacture at large scale. And in the case of this particular platform technology, we knew the dose that we wanted to use, and we knew the likely side effects that we would see from vaccination in the majority of subjects because we'd used the platform so much before. So the vaccine is based on a non-replicating simian adenoviral vector vaccine, which is called ChAdOx1. So it's based on an adenovirus, which is a virus that commonly causes infections in people. There are lots of different adenoviruses, each with a different number after their name, and they give us a mild infection, so essentially a cold. And we can use this virus to make a vaccine and to deliver the cargo of the genetic information to produce a protein taken from another virus, which in this case is the spike protein of the novel coronavirus, which is why it was nCoV-19 originally. Now, of course, that virus is known as SARS-CoV-2. But we need this vaccine to be safe, and we don't want it to spread through the body after we use it to vaccinate somebody in the way that the adenovirus normally would. So we make it into a non-replicating viral vector by removing some of its genes. That means that the adenovirus can still infect cells in our body after we vaccinate somebody, and it can make the spike protein inside those cells, but the adenovirus can't then make any more of itself, and it can't spread through the body any further. So even in people who fail to make an immune response against the adenoviral vector, they won't suffer an ongoing infection with the adenovirus itself. So we've taken out some of the original genes and we've put in the gene to encode the spike protein, and then we can manufacture this in large quantities and use it as a vaccine. And because it's a virus and it's live, even though it doesn't replicate, it's very good at interacting with the immune system. So what we want a vaccine to do is two different things. First of all, we need to deliver the antigen, the protein that we want to make an immune response against. So here again, the spike protein of the coronavirus. But we also need to inform the body that this is something that the immune system needs to respond to. So we need danger signals. We don't want to just put a large amount of the spike protein there and have the immune system ignore it. And because it is actually a very short-lived viral infection, immediately after vaccination, that provides the body with the danger signal it needs, and then it looks for what's new in the body, and what's being produced in large amounts is the spike protein of the coronavirus. So we get a very good immune response against that spike protein. We get antibodies and we get a T-cell response as well, and that helps maintain the response so it's long lasting. And we'd been developing this technology for some years before 2020. And by April of 2020, we'd already worked on 12 phase 1 clinical trials. So that's vaccinating people with ChAdOx1 expressing antigens taken from different pathogens, not just viruses, but also bacteria and malaria, which is a parasitic infection. And we'd vaccinated 330 people with ChAdOx1 vectored vaccines, but more broadly, other simian adenoviruses, so other ChAds, had been used in over 6,000 people of all ages, from infants up to people in their 80s and in different parts of the world. And we saw a consistent safety profile and strong immune responses after one dose. We'd also made a vaccine against another coronavirus, Middle East Respiratory Syndrome, or MERS. We'd vaccinated non-human primates with that vaccine and then exposed them to live MERS virus and shown that they were protected by the vaccination. And that vaccine had also been in clinical trials, both in the UK and in Saudi Arabia. So if I move to the next slide, this is some of the data that we generated in that MERS clinical trial. So just to orientate you, on the top left, we're looking at antibody responses. And in this trial, we gave 3 different levels of dose of the ChAdOx1 MERS vaccine. So group 3 had the highest dose, and group 3 resulted in people generating the strongest antibody response. It's strongest at 28 days after vaccination. That's exactly what we'd expect. And by 6 months after vaccination, it has declined. And again, this is normal. But a year after vaccination, there's no further decline. So this is what we would expect to see with any vaccination. And it's important that when we come to think about waning immunity to SARS-CoV-2, if we're talking about antibody levels declining after vaccination, this is completely normal. This is what we would always expect to see. So the peak a month after vaccination, then a decline, but it doesn't decline all the way back to baseline. It's maintained at a higher level than it was prior to vaccination. And that's true even after one dose. And in the middle at the top, we're looking at the T-cell response. So cells in the body that are able to recognize the antigen, which in this case is the spike protein of the MERS virus, and they would then be able to destroy any cells that are infected with the virus and prevent its spread in the body. So again, we're seeing that we are inducing T-cell responses by vaccination in all the groups. They're shown here combined. T-cell response peaks at 14 days after vaccination. That's normal. Again, it declines. Again, it's maintained above the initial level a year after vaccination. But the T-cell response isn't completely negative in our vaccinees before we vaccinate them, because there are 4 seasonal human coronaviruses that infect us at multiple times throughout life. And some of the T-cell responses cross-react with the MERS virus and also with the SARS coronavirus. So that's why we see a low-level T-cell response even prior to vaccination. And then on the right-hand side, you can see where we're measuring neutralizing antibodies against, in this case, the MERS virus. We want to see if antibodies can bind to the MERS virus and prevent it from being able to infect a cell, which is what characterizes a neutralizing antibody response, and we can see that we are inducing neutralizing antibody responses more so with the highest dose group, so group 3, than in groups 1 and group 2. And on this slide, we're testing it against a number of different isolates of the MERS virus, but in both cases, the highest dose that we gave resulted in the greatest induction of neutralizing antibodies. And so this information was very useful when we came to planning clinical trials with the SARS coronavirus vaccine. We went straight for group 3, the highest dose level, because we knew it would give us strongest immune responses, particularly when we come to think about neutralizing antibodies, which are the ones that we are most interested in inducing to get protection against infection. So we had previously been working on vaccines against outbreak pathogens. So there's a list that the WHO and other organizations had generated of viruses that have caused outbreaks in the past and for which we don't have any vaccines. These are known viruses. There are things like Ebola virus, Lassa fever, Rift Valley fever virus, Crimean Congo hemorrhagic fever virus. Some of these viruses can infect domestic livestock, such as cattle, sheep, and goats, and from there they can sometimes infect people. Some of them are found in bats or mice and can directly infect people from there. So Nipah virus is found in bats in Asia, and occasionally when the fruit bats have been dropping contaminated fruit and somebody picks it up, they can cause Nipah virus infection in a human. So the focus had been on developing a panel of vaccines against known viruses, but we also were encouraged to think about what would happen if a new virus was identified, and we quickly needed to develop a new vaccine against a virus previously unknown. And we had done some planning, at least some thinking about that, but we hadn't really had the opportunity to put those plans into practice. And then we suddenly needed to do that in January of 2020 when we first started hearing about the novel coronavirus. So in summary, what we needed to do was go faster and go bigger. We needed a pharma partner because although the university can do some vaccine manufacturing and run clinical trials, we are never going to be manufacturing at the scale that's needed for market supply. And we need a pharma company to partner with us to take on that activity. We needed to plan for the future and plan many steps ahead from the beginning so that we could shorten the timelines to do all of this. And that meant that we were continually working at risk. But that's not at risk to the safety of the people who receive the vaccine. It's at risk to the money. So normally, when we do the first stage of manufacturing a new vaccine, then we would reach a certain point, stop, arrange for a battery of tests to be completed, get those test results. They all need to be passed. We would review that and then continue with the next stage. In 2020, we didn't stop and wait. We continued with the next stage of vaccine manufacturing in parallel with getting those tests completed. So we still had all the same tests and all the tests were passed, but we didn't wait to get the successful test results before we moved on with the next stage of manufacturing. And that means we're putting money at risk, because if any of those tests have failed, we would have had to throw away what we'd started making, go back and start again. That never actually happened. We have a very good manufacturing team and we never fail the tests, but we were working in a different way in order to make sure that we could move on as quickly as possible. And all of the test results from all of the stages of manufacturing were completed and presented to the regulators, the MHRA, before we could start the clinical trials. But they did a rolling review, and so as each piece of data came in, they were able to review that and give their opinion, so that by the time we got to the last test results coming in, we were ready to start the clinical trial very rapidly. So we did start our clinical trial in Oxford on the 23rd of April of last year, and we ended up with 1,077 people in this trial, which is quite large for a phase 1 study. Half of them had the COVID vaccine, half of them had a control vaccine, a meningitis vaccine, so that they didn't know what they'd had. If we'd given them saline as a control vaccine, they might have worked out that they weren't having any reactions to it, and they might have known which group they were in, and we didn't want that to happen. So again, we're generating the same sort of data on the bottom left, neutralizing antibody titers. And we decided with a small group that we would test administration of 2 doses of the vaccine rather than just 1. And we saw that we did get an increased neutralizing antibody titer after a second dose when it was given 4 weeks after the first. At this point, we didn't know how high the neutralizing antibody titer would need to be to protect people. So having seen that we could increase it with a second dose, we then changed the design of the trials to incorporate a second dose into all of the trials. The interval, however, was not always 4 weeks because we didn't always have the vaccine supply available to do that. We're also measuring what we call the adverse events after vaccination, which unfortunately on this slide you can't see any great detail, but what we're looking for here is, do people have a fever? Do they feel feverish? Do they have an aching arm? Any other common adverse events that we expect to see after vaccination. We record all of that in all of the participants and put that together, and we saw slightly more reactions to the COVID vaccine, to the meningitis vaccine, but all was very much as we'd expected from previous trials, and we saw that antibodies were being induced and that some of those were neutralizing antibodies, and we also saw T cells being induced. So we were then able to move to the next stage of our trial, a phase 2 trial, and this is where we start to broaden the range of participants in the trial, in particular increasing the age range of people. So the phase 1 trial was people aged 18 to 55. We now start to recruit people in 2 different age groups, 56 to 69 and over 70 years of age, because we need to know about the performance of the vaccine in all adult age groups. And from there, we then moved on to further clinical trials. And this then really started to expand. So at the same time as starting our phase 2 trial, we started our phase 3 trial in Oxford, first of all, in the younger age groups. And this is where we have a much larger number of people enrolled, half of them getting the COVID vaccine, half of them the control vaccine. And then we follow them up and we ask them to report to us if they have any symptoms of coronavirus infection, and we check to see if they're PCR positive. And over time, we are able to look at the numbers of people in the 2 different groups, the COVID vaccine and the control vaccine, to see who's been infected, and that allows us to calculate the efficacy of the vaccine. Trials also started in South Africa and Brazil, sponsored by the university, because it's important to look at the performance of the vaccine in different geographical regions. And when AstraZeneca joined us, they also started to sponsor clinical trials themselves, particularly in the US. But also with smaller trials in India and Japan. And so we were getting data from many different places in the world. AstraZeneca also very quickly built a supply chain network to manufacture the vaccine at scale. They didn't wait to find out whether the vaccine was effective or not, because if they had done, and we discovered that we had an effective vaccine, we would then have had a long delay before having any vaccine available to use to prevent illness in people. So they started this activity very, very early, and they've worked with more than 25 different organizations over 15 countries to have large-scale manufacturing enabled in different geographical regions, and the aim of that was to have local supply around the world. So coming back to the clinical trials, we need to define what we're looking for as the primary outcome. And in our trials, it was symptomatic PCR positive COVID-19 with at least one other symptom of COVID-19, so not asymptomatic infection. And to count as an endpoint in the trial, it had to be at least 15 days after the second dose of vaccine, and the people who were experiencing this infection had to have no evidence of antibodies present prior to being vaccinated. So in the first analysis, we only included data from 2 of the clinical trials. So we had 2 trials in the UK, COV1 and COV2. COV1 was the phase 1/2 trial, and COV2 was the phase 2/3 trial. And COV5 was the trial of 2,000 people in South Africa. Whereas the COV3 is the much larger trial in Brazil. And to be included in the first analysis, there had to be at least 5 symptomatic PCR positive cases in each of the trials, and the 2 of the trials didn't meet that, so we only included data from the other 2 trials initially. So one thing we wanted to know about was, is the vaccine effective at preventing hospitalization and severe COVID? Because as we're now very much aware, if somebody gets a COVID infection which is very mild, that doesn't really affect healthcare systems. What we want to know about is the ability of the virus to make people seriously ill and require hospitalization, because then we're thinking about the effects on healthcare systems. So what we saw is that from anybody who'd got to at least 14 days after their second dose of vaccine, there were no hospitalizations in the COVID vaccine group, whereas there were in the control group, and there were no cases of severe COVID in that group, which was a very good outcome. But then we also reported on the efficacy of the vaccine to prevent symptomatic COVID, not necessarily severe or requiring hospitalizations. And here, the first readout showed us that the vaccine was 70% effective at preventing symptomatic COVID disease. So this actually was slightly more complicated than the 70% in everybody. And when we broke this down into different groups, we could see that there was one group who'd had a low dose of the vaccine initially, followed by the full dose, who actually had a high level of efficacy. And it took some time to understand this, but actually it turns out that it's not the dose level that's important. What is important is the gap between the two vaccinations. And we subsequently generated more data to show that a longer interval between the vaccinations increases the antibody titers and increases the efficacy of the vaccine. So a long interval is a good thing to have. So we then got our approval to use the vaccine from the MHRA at the end of December last year. And the approval was to give 2 doses 3 months apart, recognizing that a 12-week gap between doses increases the antibody titers and increases the efficacy of the vaccine. And that's what informed the vaccine rollout in the UK. We were then able to provide more data from the clinical trials, which didn't stop when the vaccine was approved. And this looked at, first of all, the efficacy of a single dose over different lengths of time. And we were able to show that up to 90 days after vaccination, there was 70% efficacy of preventing symptomatic COVID disease. And so there was no disadvantage to people in waiting 90 days to have their second vaccination, because the efficacy didn't decline in those first 90 days. We then looked at the efficacy of after the second dose with different intervals between the 2 doses. And you can see in the final column the vaccine efficacy, which increases as the interval between the 2 doses moves from under 6 weeks to more than 12 weeks. So having at least a 12-week interval between those 2 doses is increasing the vaccine efficacy. It also meant that more people could be vaccinated with their first dose, while people were then waiting 3 months to have their second dose. And it results in good protection after the first dose and then an improved level of protection after the second. So those were all data that came out of our clinical trials. But what we then started to see outside of the clinical trials is what's known as vaccine effectiveness data. And this is when the vaccine is used in the general population, and Public Health England monitor the effectiveness of the vaccine by measuring who is being hospitalized and measuring deaths in the general population. And they were quickly able to show us that in the over 80s, which was the population that first received the vaccination, the vaccine was reducing hospitalization by 80%. And this was true of the AstraZeneca vaccine and also the Pfizer vaccine. And we could also see, if looking at the graph on the right-hand side, that deaths in the over 80s were reducing faster than in the younger age groups. And that was a combined effect of both of the vaccines. And that's because the over 80s were being vaccinated first. And so we were seeing a rapid reduction in deaths in that age group who then started to be vaccinated. So that was due to the regulatory approval of the vaccine in the UK. But AstraZeneca worked very hard to get regulatory approval to use the vaccine in many other parts of the world. So the EMA, European Medicines Agency, approved. WHO gave their pre-qualification approval, and that means that many countries will be able to use the vaccine because of the WHO approval. And they continued to get approval to use the vaccine under the various different mechanisms for emergency use or conditional approval or special import authorization that exist in different parts of the world, because we had always wanted this to be a vaccine to be used very widely around the world. It's a vaccine that doesn't require frozen storage. It can be stored in a normal refrigerator, and that means it can feed into existing supply networks that are used for other vaccines and makes it much easier to use than vaccines that do need to be stored at frozen temperatures. AstraZeneca had also, as well as agreeing to make the vaccine available in very large numbers of doses, they are making it available not for profit during the pandemic and for low-income countries even after the pandemic ends. So this was realizing our aim of making a vaccine for the world, and part of that, a very important part, is to have regulatory approval across the world so that the vaccine can be used in many different countries. AstraZeneca also worked very hard on their vaccine manufacturing network, setting up manufacturing in many different countries across the world. For example, in Australia, a company that would normally make flu vaccines is now making the AstraZeneca vaccine so that it can be supplied in Australia from that manufacturing site without having to ship it around the world. And we have a partnership with Serum Institute in India, who are the world's biggest vaccine manufacturer. They normally make the largest number of doses of other vaccines, and they are now making very large numbers of doses of the COVID vaccine. Moving on, we started to see variants of the SARS-CoV-2 virus arising, where the spike protein is different, and the antibodies that we get from vaccination don't bind quite so well. They don't neutralize quite so well as they did with the original virus. But we need to know if the effectiveness of the vaccine is being changed by that. And again, Public Health England have been able to generate this data. And what we're looking at here is the vaccine effectiveness against hospitalization, so preventing people from being admitted to hospital with COVID disease, of both the Alpha variant and the Delta variant. And of course, it's now the Delta variant that is predominant in the UK and pretty much everywhere else in the world. So after 2 doses of the AstraZeneca vaccine, we're seeing 92% effectiveness against hospitalization with the Delta variant. That's across all the age groups. So that's still a very good result. And if we look instead against the ability of the vaccine to prevent symptomatic disease with the Delta variant, it's 86% with the Pfizer vaccine and 67% with the AstraZeneca vaccine. So we're still seeing good effectiveness against preventing symptomatic disease. What matters more is the ability to prevent hospitalization, and that's still working well even against the Delta variant. We've been able to generate more data from our clinical trials that shows that with an even longer gap between the 2 doses of the vaccine, we see even higher antibody titers. So the group here who had the 45-week interval between their 2 vaccine doses end up with the highest antibody titers after the second dose. That's useful information to have. So if people have been able to have a first vaccination and then there's a delay in getting the second vaccination to them, they will actually benefit from that gap. And it doesn't mean that they need to immediately have a third vaccination because they'll have a very good response to their second vaccination. But if we do give a third dose, we get even higher levels of antibody titers. There had been some concern that because we're using an adenoviral vector, that giving it second time, a third time, we would actually not see any increase in antibody titers because of immune responses generated against the vector itself. But that's not true. If we look at the antibody titers here, and I wish I could point, but unfortunately I can't do this when the slides are being shown remotely. But if we look at the 28-day time point after each vaccination, we can see it increases from the first to the second and the third. So we see improved neutralizing antibody titers after each individual dose of the vaccine, and that applies to variant viruses as well, because we tested neutralizing antibody titers to the beta variant, which is the one that's most different from the original virus, and we see increased antibody titers to the beta variant as well after a third dose. So that's good news. Now things have moved on to look at heterologous Primeboost vaccination regimens. So this is giving different types of vaccine, a Pfizer vaccine followed by an AstraZeneca, vice versa. And what we're seeing here is that all of the combinations result in increased antibody and T-cell responses after the second vaccination, and that the safety is good. And so these different combinations can be used. And there are more combinations being studied in a further study called COVID-Boost, from which data has not yet been released, but that should be coming shortly. So this is not trying to find the ideal vaccination regimen. This is trying to find out what is possible to use, because around the world, different vaccines may be available at different times, and we want to know if people have had one vaccine but then a second one is available, is it appropriate to give them a different vaccine for the second vaccination? And so far we're seeing that all of these combinations look very encouraging. We're also working on updated versions of the vaccine with variant spike proteins. So as I said, the beta variant spike is most different from the original spike. Delta is not quite so different. And we don't know if we need to change the spike protein that's in the vaccine, so we're doing some experiments to test that. So we've produced Channels-1 expressing the beta spike production, the beta spike antigen, and have generated some data in preclinical studies, which is shown here. So this is immunizing mice. And what happened was that the mice were given a first dose of the original vaccine, which is the AZD1222, and then a second, either nothing the second time or the beta variant in the second vaccination. And we can see, if we look at the neutralizing antibodies against all of the variants, so the original virus, the beta, the kappa, and the delta variant, we see that we improve the antibody response against all of the variants with the second vaccination, which is the beta variant spike. It doesn't just improve antibodies against the beta variant, it also works well against the original virus, the kappa virus, and the delta virus. So that's good news. Alternatively, if we compare 2 doses of the original vaccine to 1 dose of the original followed by 1 dose of the beta variant, we still see good antibody responses with both of those approaches to vaccination. And again, it applies across the variants. So those data were generated in mice. There is now a clinical trial happening with people who've had 2 doses of the original vaccine and then get 1 dose of the beta variant vaccine. And that's happening in the UK. And then in some other countries where people had not already received vaccines, they're getting just 1 dose of the beta variant. So we'll be able to see in a few months' time what happens to their antibody responses and decide if there is any need to change the vaccine that's in widespread use. But so far, as in the mice, we see very good responses to beta and delta variant, and we've also seen very good vaccine effectiveness against the delta variant just from using the original vaccine. So although we have a process for making new versions of the vaccine should we need to, at the moment it doesn't seem as if there's any particular need to do that. We will continue to follow variants as they arise and make new versions of the vaccine if any variant looks to be particularly different, and we will then assess if it needs to be taken into production. So I'm going to stop there, but I can't stop without acknowledging the help of so many people that have been important in generating all of this data. First and foremost, the volunteers that came forward to take part in our clinical trials in very large numbers and have been very patient with us in coming back and regularly giving us samples of their blood so that we could see what the vaccine was doing to them and enabling us to generate the data that means the vaccine can then be licensed and used widely. The work was initiated in my group at the Jenner Institute, but we very quickly joined forces with the Oxford Vaccine team, Oxford Vaccine Group, run by Professor Andy Pollard in the Department of Pediatrics, who took on the running of the clinical trials. We worked together on generating the immunology from those trials, but we've also worked with 18 other clinical trial sites around the UK and with excellent clinical trial sites in Brazil and South Africa. And all of those trial sites have been running incredibly efficiently and allowed us to conduct these trials very quickly, but also to extremely high standards, generating data that we can use for the regulatory approvals. And of course, I'd like to thank our funders and our partners in this work, AstraZeneca, who took on a very large program of work and really invested heavily to make sure that we could have enough vaccine to supply around the world. And now more than 1.3 billion doses of this vaccine have been released. Thank you very much.
Thank you so much, Sarah. That was absolutely fascinating. We've got some questions now from the audience. So thanks to our audience for sending in questions in advance. And I'm going to read them so that because I'd quite like to be true to the questions as they were asked. So I've got some questions about how the vaccine works. I've got some questions about vaccine development and rollout. And I've got some questions about working as a scientist. So the first question from the audience relates to vaccines for the variants of the virus. And the question asks whether there are any practical limits to the editing of the COVID vaccine to track variants of the virus, and whether it's possible to assess the likely interval during which a version of the vaccine will remain effective as the virus changes.
Well, it's very easy to make a new version of the vaccine with a different spike protein in it, and we've already been doing that. We started actually in December of last year. We realised that we were going to need to respond to these variants, and we now have a pipeline in place to go from identifying a new variant all the way through to clinical trials of that new variant virus, and we're doing that with the beta variant, and that was chosen because it's most different from the original virus. It's not really possible to predict which of these new variants we're going to need to make a new vaccine against and start using it. And there are practical limitations, not in our ability to generate new versions of the vaccine, but the logistical difficulties in manufacturing different versions of the vaccine and then having those replace the original version. And what we certainly don't want to do is to get into a situation where we are looking at using different versions of the vaccine in different parts of the world to counter different variants. And it doesn't look like that would ever make sense, because we've seen that a successful variant such as Delta actually spreads all the way around the world. And so the problem is the same everywhere. So really, we can be most efficient in our vaccine production if we minimize the changes. And the approach we're taking is to be prepared to make a change, but only do that when it really becomes clearly necessary.
Thank you very much. As a follow-on from that, one question asks whether you ever had any doubts that a suitable COVID-19 vaccine would be developed at all, or whether it might take many years to do so.
Well, I knew before we started that there are successful coronavirus vaccines that are used in livestock. In chickens and in cattle, there are coronavirus vaccines that are licensed and that are used, and they're effective. We don't have any coronavirus vaccines against any coronaviruses that infect humans. We've been starting to work on the MERS vaccine. Initial work on SARS-CoV-1 never got very far. So some people doubted the ability to make a vaccine against coronaviruses, saying that it hadn't been done before, but in fact it has been done before for livestock vaccines. And we'd also seen good responses in our MERS clinical trials. So I never doubted that we would have a vaccine. The question was always going to be what level of efficacy we would get with that vaccine and how many doses we would need to achieve a useful level of efficacy.
Thank you. And then again about the vaccine itself, there's a question about sequences within the vaccine. And the question reads, are there any particular sequences within your vaccine that are particularly immunogenic? And if there are, then might a future vaccine avoid these?
Well, by far and away, the most immunogenic sequence is the spike protein, the antigen that we want to produce. And we've shown, with the help of one of the researchers at Bristol, actually, David Matthews, has helped us show that the vast majority of the protein that's being produced after vaccination is the spike protein, or whichever other antigen we want to express. We don't get very much adenovirus protein expressed. So therefore, we get most of the immune response against the right protein. But for the adenovirus itself, the major protein around the adenovirus is called the hexon. And so different serotypes of adenovirus have different hexons, and that's why antibodies to them don't always cross-react. So if we did want to modify the adenovirus, it would be the hexon protein that we would need to look at. But actually, an easier thing to do is probably just to start from a different serotype of adenovirus, which already has a different hexon. And we do have a second simian adenovirus called ChAdOx2, which is derived from a different starting point, and so its hexon is different.
Thank you. And then we have some more general questions about how it was to develop the vaccine and roll it out. The first question asks what the most challenging aspect in developing the vaccine was.
Initially, the most challenging aspect was trying to get some money to get the program underway, and then some opportunities to apply for funds became available. And by the time we'd been awarded those funds, we'd already spent them and needed a lot more money to move to the next stage. So that was difficult initially. That got a lot easier by the end of April, when the vaccines task force had been formed and decided to invest in this vaccine. That meant that we had the ability to move on with the clinical trials and expand them. I think the other really challenging thing was trying to plan many steps ahead all of the time. There's a lot of experience with the people that I work with in doing early vaccine manufacturing, production, clinical testing, but we don't normally do it so quickly. So between all of us, we had to put together plans to move from the concept to phase 3 trials and onto vaccine licensure within a year, which is much faster than we'd normally do it. So we were asking ourselves questions about what we're doing today, what we're doing next week, what we're doing next month, and where we want to be in 3 months' time, all at the same time, and continually doing that as we develop the program, which was quite a challenging thing to do.
So we've had some questions about exactly that, about the rapid development of the vaccine. And people are really interested to know what we've learned from the process that could be used to inform future vaccine development. So what might be done in the future leading from this experience?
Well, we've certainly seen that platform technologies enable us to move quickly, because, as I said, we do so much of the development on the technology itself that it reduces the amount of work that has to be done on a specific vaccine. So RNA vaccines are a different platform technology, and the same applies to that platform technology. Some of the things that we did in 2020 won't necessarily be applicable to future vaccine development, because the big thing was that we were all working on the same vaccine. The regulators were prioritizing it, and they can't prioritize multiple things all at the same time. So the rolling review that the regulators did was very fast, but from their point of view, it's not particularly efficient, because they had all of their team working on one project. So I think there are things that we can learn about in overlapping programs. If we're prepared to put money at risk, we can move more quickly. That's often not possible in academic setting. Companies are more likely to do that. And using the platform technologies really does save a lot of time.
Thank you very much. Hope for the future, I think. So then thinking about kind of vaccine programs around the world, some people were interested to know what you think could be done to quickly and effectively vaccinate the population around the world, especially in the global south.
So the good news on this point is that the number of doses being manufactured and released for use each month is increasing. And the number of vaccines that are being licensed for use is also now increasing. So as each month goes by, we have more different vaccines to use and more doses of the original vaccines available. So by the end of the year, it's projected that about 11 billion doses of all of the COVID vaccines will have been made. And the challenge, I think, is going to move from being able to make enough doses available to being able to make those doses available in the vaccination clinics around the world, because we need to not just get those doses into particular countries. Once it gets into a country, it needs to spread around the country and be transported so that it's ready for use in all the different areas of that country. And that's where there are going to be some challenges now that need to be solved next.
Thank you very much. Sarah, we also received some notes of thanks to you for your work. And some people asked what motivates you to do your science?
Well, I've always been interested in using science to create interventions to improve people's health. And vaccines are a great way to do that. They're the most cost-effective public health intervention, and they can have really broad impact. I've worked on the development of a number of vaccines against the outbreak pathogens for a while. It's been quite slow, can be quite frustrating to move at such a slow speed. And so when we had the opportunity to work on a virus that turned out to have such broad impact across the world, and put the science that we'd all been working on as a big team in Oxford into practice, we all jumped at the opportunity. We didn't realise at the beginning how much work it was going to be and how it was going to completely dominate our lives for such a long period of time. But we were very glad to be able to have an opportunity to use what we've been developing for so long to actually put it into something that would make an impact.
Thank you. And a very last question. We have some school students listening in live today. What advice would you give to school students who want to become scientists?
I think the most important thing is to find the bit of science that really makes you tick. There are lots of different ways of working as a scientist, and there are lots of different aspects of science. So some of the people that I work with spend their entire working lives working with data on a computer, and some people at the other end of the scale spend their lives working with volunteers coming into vaccination clinics and talking to them and working with people all the time. So there's something for everybody in between those two extremes, and there's also maths, physics, chemistry, biochemistry, biology, lots of different areas. So I think find which bit really makes you tick and then follow that. But don't make your options too narrow too early. Don't specialize. Wait until you get to university and do a bit of all the different sciences if you can, and then decide where you want to go from there.
Sarah, thank you so much. I think that's a great note of inspiration for us to conclude the questions and answer session. Folks, thank you for sending in your questions. Sarah, thank you ever so much for answering them. I'm sorry that we didn't get a chance to ask all of the audience questions, but hopefully you feel we've covered a good breadth of ground in the Q&A session. Sarah, it remains for me to thank you enormously for giving the 2021 Elizabeth Blackwell Institute lecture, and to thank you. We've sent you a gift, and the gift is traditional Bristol blue glass, which was supported by a generous donation to the Institute from the descendants of Elizabeth Blackwell herself. So, Sarah, I know you have the gift with you today.
Thank you. Yeah, it's a beautiful blue glass vase engraved with the date of today's lecture, and I will treasure this and keep it in my office. It will become part of my background. Thank you very much.
Thank you so much, Sarah. Thank you. It was an inspirational talk today. I think we've all learned a lot. Thank you very much, everyone in the audience, for joining us wherever you are. If you'd like to watch and to listen again, or tell others about the event and tell people who would like to listen to Sarah's talk and see her presentation, we'll be making the video publicly available from the start of next week. And you can find it on our website, which is www.bristol.ac.uk/Blackwell. And thank you again, Sarah. That was absolutely inspirational, and we're enormously grateful that you found time in your busy schedule today to do this lecture live. Thank you.
Thank you very much.
Thank you and goodbye.